We're moving now on to Juvena Therapeutics or Juvena Therapeutics. We've got Hanadie Yousef, co-founder and CEO. Hey, Hanadie.
Hi, how's it going?
Yeah, great. So very interested to hear your presentation. Are you ready to roll?
I am. I will get into presenter mode.
That's perfect. Awesome. Over to you.
Great. Well, thank you so much for the invitation to present today at the Emerging Company Showcase. It is my second consecutive year participating in Metabesity, and I found it to be a very engaging and informative conference, really bringing a lot of movers and shakers in the health span and longevity biotech space together. So I really appreciate it. At Juvena Therapeutics, we are dedicated to ultimately developing protein therapeutics that can promote tissue regeneration and repair in order to rejuvenate lives. And ultimately, the reason we're so passionate about this is because we see, as we all know in this conference, a looming healthcare crisis. By 2050, it's estimated by the World Health Organization that there will be over 2 billion people worldwide over the age of 60, and it's aging itself that is the greatest risk factor leading to multiple comorbidities, chronic degenerative diseases, in which there are very few, if any, FDA or regulatory approved therapeutics on the market that can actually target the underlying mechanisms causing the disease to reverse and even repair symptoms, as opposed to just treating symptoms. And so at Juvena, we are ultimately focused on really targeting systemic and local signaling pathways that are deregulated with aging to restore cellular homeostasis. So during the aging process, many signaling pathways, both locally in the tissue niche as well as systemically, are deregulated. You have, for example, increase in many inflammatory pathways and cytokines caused by increase in senescent cells and secretory profiles. You have, reduction in many regenerative growth factors and signaling proteins systemically and locally in tissue. This leads and really drives a lot of cellular dysfunctions, such as altered cell communication. Really, it begins to inhibit stem cell function and really prevent stem cell mediated tissue regeneration and repair. You get mitochondrial dysfunction, loss of proteostasis, and other really key hallmarks of aging that results in disease manifestations, from neurological diseases through to metabolic, muscle wasting disorders, articular damage, cardiovascular, and more. So at Juvena, we're really focused on manipulating these pathways to really restore them to more youthful homeostatic levels to promote very naturally tissue regeneration and repair. And we're doing this by harnessing what we really consider to be the most pro-regenerative source on planet Earth for people, which are human embryonic stem cells, and specifically the proteins that they themselves secrete to promote development of every tissue in the body. My colleagues and I, during my time, my PhD studies at Berkeley, really observed and characterized that by isolating a subfraction of proteins that are produced and secreted by human embryonic stem cells, and then figuring out a way to kind of concentrate and purify them, when we added them onto a variety of either primary aged human muscle precursor cells in vitro, or even injected them into a variety of animal models, we could really enhance these regenerative phenotypes. So for example, these proteins can really promote better skeletal muscle regeneration and reduce fibrosis in aged mice when injected intramuscularly. They can also really enhance age or diseased human myogenesis. These proteins, when added onto aged primary human chondrocyte precursors that are isolated from osteoarthritic knees, can promote their survival and better chondrogenesis. And when added onto human cortical neurons in the face of A beta, they can prevent their apoptosis, really promote survival. So it's these regenerative phenotypes that are already countering phenotypes associated with aging that got us excited and got me excited to really develop a systematic method of actually characterizing these proteins and translating them into therapeutics for people. So ultimately, at Juvena, we're developing protein therapies that can activate regenerative signaling pathways deregulated with aging in order to delay and even reverse degenerative diseases. And we're doing this by really systematically discovering regenerative proteins secreted by human pluripotent stem cells. And we're doing this by a very systematic method. I'll get into the detail in a moment. And then engineering them, once we really show efficacy, into biobetter versions of themselves as novel protein-based therapeutics for tissue-specific degenerative diseases. For example, by engineering them as fusion proteins with better pharmacokinetic and dynamic properties. And so we're doing this through having established a multimodal machine learning enhanced protein drug discovery and preclinical development platform that is capable of identifying novel regenerative signaling proteins from the secretome of human stem cells that can rejuvenate any tissue type, and then translating them into potent protein-based therapies. And we do this by really combining multiple, data modalities, including really state-of-the-art proteomics, and really using proteomics to unravel and identify and characterize secretomes of stem cells, combined with transcriptomics and image-based high-throughput screening of primary human donor-derived precursor cells with machine learning and computational techniques in order to ultimately discover and then develop novel regenerative proteins as therapeutics for regeneration. Our internal proprietary data sets and database is also augmented, really improved by really also taking advantage of many publicly available human and mice omics data sets that have been recently published, with the just tremendous kind of advancement in sequencing technologies and proteomics and at the transcriptomics level, leading to many wonderful publications and now data sets on aging that us and many companies can really take advantage of to mine and for target identification. And so really our advantage at JuvNET is having developed and continuing to build up a unique platform-enabled approach to identify novel pro-regenerative proteins. So we start by isolating and really characterizing the secretomes of human pluripotent and multipotent stem cells. We then take these proteins and screen them for regenerative phenotypes, really focused on the phenotype-based screens and targeting, phenotypes that we know are key drivers of the degenerative processes due to biology of aging. So for example, identifying and characterizing proteins and the secretomes that can promote differentiation, that might be immunomodulatory, antifibrotic, or even anti-apoptotic. And then once we identify and really characterize these secretomes, we deconvolute them using unbiased mass spec-based quantitative proteomics to really identify what are the key drivers of these phenotypic regenerative outputs within the secretomes. Once we do that, we then move on at JuvNET to really do an AI-enabled rank ordering of proteins within the secretome, using both bioinformatics, computational techniques, as well as various deep learning models that we've established and collectively dubbed JuvNET as our toolkit to really assist us in identifying, rank ordering, and predicting what proteins will work individually or in combinations of 2 to 3 to really mimic what the secretome as a whole is doing for a given tissue and cell to promote a regenerative phenotype. Once we have those assays in vitro, we then really move on, or actually I'd say through the in silico screening, we move on to perform high throughput human cell model screens, followed by really extensive preclinical animal model validation, using most importantly natural aged mice as a model to rejuvenate tissue function, but also really honing in on disease specific models and preclinical models to identify many candidates. And then of course, once we have those hits, we move on to at JuvNET perform lead optimization. So this in the last 3 years and few months of our full-time operations has really led to, as our first tissue target, we've been really focused on muscle rejuvenation. And so we have several candidates that can really enhance not just better myogenesis in vitro, but importantly, improve strength and function and muscle regeneration in vivo, and could be used to treat a variety of muscle wasting diseases, with a lead program really poised for a rare autosomal dominant muscle wasting disease known as Myotonic Dystrophy Type 1, which I'll share about in a moment. And importantly, a platform poised for really growth and expansion and identification of protein therapeutic candidates for a host of tissue-specific degenerative diseases. And we are currently raising a Series A in order to really advance the platform and pipeline to really fill and identify numerous protein candidates for several new programs, as well as to advance our lead program for muscle wasting. And so these protein therapeutic candidates were identified and screened and developed based on our first proprietary library we established, which was identifying really key drivers of rejuvenation in a subfraction of the secretome of human embryonic stem cells. So we did a variety of experiments really utilizing my co-founder, Dr. Jeremy Connell's expertise in mass spec-based quantitative proteomics to identify proteins that are, for example, secreted by hESCs that can induce regenerative phenotypes, but the same exact subfraction of proteins that are produced by differentiated cells don't elicit a regenerative phenotype unaged human precursors and cell models. And so using that kind of differential combinatorial analysis, we really honed in and discovered about 1,000 proteins, signaling proteins that are enriched in the secretome of hESCs that are really responsible for driving regenerative phenotypes for multiple tissues that we've tested. And we do plan to expand the library to multiple multipotent stem cell secretomes upon closing our Series A financing and gaining the financial support to do that. And so ultimately, this is kind of another schematic to show our process where we take our library and really integrated every step in the process, machine learning and deep learning and computational techniques that really enable us to first rank, order, and predict what proteins in our library can promote regeneration for a given cell or tissue type, followed by then performing high-throughput screening using ultimately primate human stem cell assays in vitro human disease models, followed by then extensive preclinical validation. And then once we get efficacy that and in multiple hits, taking the best candidates and really performing lead optimization to turn these candidates into biobetter versions of themselves in actual potent therapeutics. And so even in our laser-like first focus on, muscle rejuvenation, we were able to screen both in mouse muscle precursors, followed by age and diseased human muscle precursors, about 100 proteins for their ability to promote human myogenesis, followed by then, and it turns out we got about 13 leads that really could either promote things like activation or even better fusion and maturation of fibers. We then tested those in vivo in several different animal models, ranging from natural aging to aged mice that were acutely injured to glucocorticoid-induced atrophy models and even muscular dystrophy models. And about 8 of those leads successfully validated in vivo in various mice models, with us then really performing thorough lead optimization on the 2 best candidates that really had robust efficacy in at least 3, if not all, models. And in fact, our top candidate really showed robust efficacy in all animal models tested, and we have now established it as a lead for a rare muscle wasting disease known as Myotonic Dystrophy Type 1. Once we get multiple hits, we then at Juvena really work on doing, and with strategy and preclinical expert consultants, a really thorough clinical and indication selection prioritization analysis of the field we're targeting, which is muscle rejuvenation, really focus on understanding the mechanism of action of which our protein therapeutics are working to identify a particular disease that we can hone in and actually develop and translate to the clinic. So with that, we really came upon myotonic dystrophy type 1, which is the most common muscular dystrophy. It's actually based on natural history studies that were conducted recently by our clinical advisor, Dr. Nick Johnson, shown to affect about 1 in 2,300 people. It's over 140,000 people in the US are known to be to have this progressive muscle wasting disease. It's caused by a CTG repeat mutation in the DP kinase gene, which leads to an RNA hairpin loop variant splicing, and then downstream of that, dysregulation of numerous critical regenerative signaling pathways that in many ways manifest as many accelerated aging. So this leads to the biggest hallmark being muscle wasting, but it's really a multi-system degenerative disease where the age of onset can vary. And, while most folks will, tend to get some muscle wasting symptoms naturally, later in life, folks with the disease will get it much earlier, and life expectancy usually isn't past your 50s. Fortunately, there are 0 FDA-approved therapeutics out there, and really Juvena has taken the lens of considering this, and it's really increasingly being recognized as an accelerated aging disease, but with an orphan disease status. And so we've also really connected with multiple foundations, including the Myotonic Dystrophy Foundation and family registry and patient registries, to really poise us for the ability to really target and support persons with this disease and improving and rejuvenating their lives and muscle function. So our lead product candidate, JV161, is a fusion protein agonist that enhances myogenesis and, importantly, muscle survival and strength when injected subcutaneously. We are targeting and activating PI3 kinase mTOR signaling, importantly with significant crosstalk and activation also of this sphingosine 1-phosphate and BMP pathways. And we have now engineered it, completed our PK studies, and really are initiating IND-enabling studies with the goal of getting an IND by early 2023 and then immediately getting into clinical trials. So just to show really a brief snippets of preclinical data, of course, we have a lot more than this, but for time, I'll just show a few highlights. Our JUV161 can really enhance myogenic transcription factors to promote better, muscle, I'd say, fusion and maturation. And it does that not just in healthy aged human muscle precursors, but also in muscle precursors isolated from patient, or I'd say persons with DM1 donors, their muscle biopsy donors. And so importantly, while our protein therapeutic, which is really, we're the only company really developed a protein therapeutic, natural protein therapeutic for DM1, while we really can enhance muscle regeneration, for example, another small molecule titled Yousef, which is in clinical studies for the congenital form of the disease to treat the autism spectrum, really doesn't do that. It's a JSK3 beta inhibitor. When injected in vivo, we can really enhance muscle regeneration, strength, and function in a variety of muscle wasting models we've tested, from natural aging, improving things like treadmill performance, grip strength, endurance, distance, also showing that it's safe and doesn't have adverse effects on things like white blood cell counts, kidney, pancreatic function, as well as using these various models listed here. And I should say, we even see robust efficacy in a muscular dystrophy model. And at Juvena, we're actually currently developing, in collaboration with a model we licensed from the Baylor College of Medicine from the Cooper Lab, a TET-inducible, really DM1 model that really exhibits a muscle wasting phenotype, given that the animal models to date don't really recapitulate the key hallmarks of the disease. And so, just to show, snippets of that data, when injected, our molecule can, or protein therapeutic can really enhance, for example, grip strength and distance run in a treadmill. In the dotted lines would be sort of the levels that you see with young, healthy animals. These are about 2-year-old mice that we use to test. We can really prevent glucocorticoid-induced muscle atrophy and really enhance skeletal muscle regeneration to levels of younger animals. We have further went on to take the natural protein therapeutic and then really develop a proprietary formulation where it's now a fusion protein agonist with significantly extended pharmacokinetic properties. And we also have, proprietary formulation of, excipients that really kind of stabilize it and boost its efficacy. And so we can, even with our fusion protein agonist, this potential developmental candidate really also show it enhances regeneration of injured muscle, can really improve weight, and then really also improve things like force measurements, the actual kind of output when injected intramuscularly. And we also see this through subcutaneous administration. And then, just even honing in on what are the pathways being upregulated, right? And is this really a therapeutic that can target the biology of aging? Even looking transcriptionally, we see multiple pathways that are upregulated that are usually based on the omics analysis, mechanistically downregulated with age listed here. We now have a candidate that has really extended pharmacokinetic half-life. So the ultimate goal would be to treat patients over a long period of time, weekly or even with biweekly subcutaneous injections, the way that insulin is administered. And we've chosen to really go after muscular dystrophy as a path to ultimately targeting large age-related diseases, where there's a clear and really rapid path to the clinic as an orphan indication with a combined phase 1/2 clinical study. And then we'll use that to really expand. And I realize I'm probably out of time here, so I apologize. Yes. We'll go after multiple pathways and targets, have an excellent team. And with that, I'll probably just end here. But thank you so much for your time and interest. And I'd be happy to take questions if there's time for it.
Great, Hanadie, thank you. So one question's come through on the chat, which is urolithin A approved by WADA, which is the World Anti-Doping Association, I would imagine?
I'm sorry, can you repeat
Phil,
that?
I think that was a leftover question for Amazon.
Oh, was it? Oh, I'm so sorry. Thank you. So, Hanadie, I've got a question for you, which relates to your funding. So you mentioned that you're just going to embark on a round. What is your timing for that, and what is the amount that you're going to be looking for?
Sure. So, we've raised about 10 million to date in seed financing, and I'm also really taking advantage of non-dilutive grant funding with, for example, the NIH. We've applied for a CIRM grant. We're raising 30 million in diligence with some folks, and definitely looking for really supportive kind of investors that really believe and are aligned with our mission and vision to build up our platform and pipeline of candidates. with really, a very kind of targeted approach to market to validate the platform.
Yeah, well, good luck with that. And that 30 million, will that all be in one tranche, or will that be in a couple of phases?
Kind of depends on how we work it out with our investors. It is a plan to be spent over about 2 years to really build up our, senior management team and folks in both the R&D and preclinical side to build up the platform and pipeline and advance our lead asset. And we, of course, have, excellent advisors supporting this as well.
Yeah, well, good luck with that, Hanadie. And I'm sorry we're so pressed for time today, but that's an excellent presentation. Thank you.
Thank you so much.
Great, thanks.